the purpose of this talk is to introduce the context of 5P medicine and how multiomics helps us to, in the end, to progress how we do medicine today and in the future. So I will start with an introduction of multiomics, because throughout this webinar, we're going to have a number of talks focused on metabolomics, but also combining different omics together. And I want to discuss the value of metabolomics, especially in these approaches. And then the main part or the main role of this presentation is really going to be to introduce the concept of 5P medicine, which may seem new to many people in the audience, but actually when I introduce it, you will probably realize this is something you've been working on for a while already. And we'll look at it through a few case studies and look into the way forward that one can take for multiomics. So multiomics is an interesting topic and something that I've been working with for the last 15 to 20 years, and that is really the passion of my scientific life. What's really interesting about it is that those omics are kind of high resolution pictures of biology at the molecular level. And what we've done is we've taken complex matrices like cells or blood or tissues, and we've separated those different molecules, whether they're metabolites or genes or RNAs or proteins. We've separated them into omic layers, as we call them, and studied them in isolation. This is what you do when you do genomics or you do metabolomics and so on. And multiomics is, of course, bringing those layers back together and then trying to rebuild the biological story that is made by those layers when they normally interact in the system that we study. And we've created this kind of increased level of complexity because we've had to develop analytical methods that allow the measurement and the quantification of these molecules. But that gives us a very interesting scientific challenge when the problem comes to bring those layers together. So for me, I find multiomic a very fascinating topic. But in this talk, I want to begin also by introducing why metabolomics specifically is an interesting omic to bring into this kind of puzzle that we have to solve. If you look at the different omics, many people know genomics. More and more people are used to transcriptomics, proteomics, even microbiome analysis or epigenomics. And more people are using metabolomics as well. What's really interesting about metabolomics is that it's very, very different from other omics in several regards. But here I want to point out the main thing for me, that is, it's very sensitive to how we live our lives. So when you're born, you have a certain genome, and that's going to give you predisposition for certain diseases. When it's monogenetic diseases, there's going to be a strong link to a single genomic trait or aspect. But when it's the way you live your life that influences your predisposition for disease, genomics is only going to inform usually like 10 to 20% of a person's risk to develop, for example, complex chronic disease of different types, whether it's neurodegeneration or certain types of cancers or autoimmune disease. There are some ties to genetics, but really we need omics that are more sensitive to the other risk factors that are diet, that are lifestyle, that are exposures to chemicals, whether they're intentional or not. And for this, metabolomics is extremely powerful. So that's why I often advocate for people, if they want to do multiomics, to combine the more established one, that is genomics, with the more the one that's closer to the phenotype, the one that is a finer measure of lifestyle, that is metabolomics, and you get very rich information already doing this. But of course, all those other omics have very specific additions that they make to the story you can build. Proteomics, for example, is really a kind of end result of how we've transcribed and translated the genome, and that can give us additional information on how those genetic differences can really impact our biology, but also together with some environmental factors or lifestyle factors that will also influence which proteins are expressed or in which quantities. The microbiome is also a very interesting one. One might think that, this is external, it's not really a person's organism, but especially from the point of view of the metabolome. When you look at what our gut microbiome and other microbiomes provide, they provide metabolites that are sometimes essential to our well-being, metabolites that we need in order to be healthy. And they can also provide metabolites that are going to be detrimental. So this is why the study of the microbiome is something that's really on the rise at the moment, and something that also works really well with metabolomics. So the white paper that you see on the right with the QR code, this is a white paper we published a couple of years ago, where we look at complex chronic diseases, so the ones I mentioned before, neurodegenerative disease, cancers, autoimmune disease. We look at them through the prism of metabolomics, and we see through the influence of food and the microbiome how we can look at those diseases through a different lens that is going to bring more than just looking at genomics, for example. So the first case study I want to put forward here is a relatively simple combination of two omics. Here we have metabolomics and epigenomics in the form of DNA methylation arrays. And I call this a simple example because it's already rather advanced to combine those omics together, but this is really the thing that you would expect when you combine those different layers. You look at a problem or a question from different angles, and you start to see different things coming up than if you just looked at them in isolation. So what we have here are metabolomics and epigenomics from post-mortem brain tissue from Parkinson's disease patients compared to control patients who didn't have Parkinson's disease. And when you look at the volcano plot on the left, what we see is, this is the metabolomic data. We see there are a number of metabolites that are higher or lower in the brains of Parkinson's disease patients. And you see, I don't know if you see my mouse, but around the middle of the orange, significantly increased metabolites, you see taurine, which doesn't look like the most drastic or interesting change because there are several other metabolites that are there, both increased or decreased, but taurine is there in the middle. And when you combine the metabolomics with the epigenomics, then you start to see taurine actually nicely associating with a number of genes that are differentially methylated and that have to do with the metabolism of bile acids. So doing this so-called simple, I mean, it takes skill to do this, but still like relatively simple combination of those omics together, you already start to see pathways or mechanisms coming up that wouldn't necessarily be the ones you would put your attention on if you just look at a single omic. And this really also kind of not confirms, but like it gives you more confidence into looking into certain pathways because you see pieces of the story in different parts of the biology that you look at with the omics. So this is the benefit for me of doing multiomics in general, but now I want to look at this in the broader context of 5P medicine. And that's an opportunity for me to introduce the topic also of 5P medicine in general, or the framework. Of course, I look at this through the prism of metabolomics because this is what I'm focusing a lot of my time on. But in the end, I see 5P medicine as a way to make the most of multiomics as well. So 5P is what it means is basically 5 words that start with the letter P and that have to do with the applications that we make today that are going to be the future of medicine tomorrow. So there's preventive medicine, predictive, precision medicine, population-based medicine, and participatory medicine. Today, I won't spend too much time on participatory medicine because just for a lack of time, but it's also a very interesting one that I encourage you to look into. But so if we look at preventive medicine, for example, this is the one where we want to prevent disease before it happens. That means we have to have sensitive measures of the risk of an individual to develop the disease, which gives us in turn innovative ways of intervening before the disease really takes place, and also monitoring whether someone is going more towards disease or towards a more healthy phenotype. And for this, metabolomics is a very nice, flexible, and sometimes reversible measure that is very, very useful for preventive medicine. Predictive medicine is one of the best known ones and also one of the best known applications of omics through the design or the establishment of biomarkers. Often these are signatures of several metabolites or several features together that form a biomarker signature. For example, for drug response prediction. So here we want to predict who is going to respond to a certain therapy, or which is going to be the best therapy to give to a specific person, which is very close to precision medicine, the third P, where in precision medicine, what we often do is stratification, or in metabolomics, we could call this metabotyping. So grouping or clustering patients based on their profile in the different ways that we measure them. You can use metabolomics or other omics to get a pattern of subgroups of a disease. I will show you an example of this, and then really have a more refined picture and also more refined options to address the different forms of the disease to really be as close as possible to what each person needs. Another one of those P's that is one that's going to be beautifully addressed by several speakers in this seminar is population-based medicine. So especially using metabolomics and other omics that are not just looking at the genetics, but looking also at the effect of the environment. In population studies, you're going to be able to look at the environmental variance in the context you're studying. You're going to be able to identify subphenotypes. And even though multiomics, I think, is relevant for all of the 5 Ps, today population-based medicine is probably the one where multiomics is best applied, and we will have a couple of examples of this, especially in the third session in this seminar. And again, participatory medicine, I will not go into too much detail today, but if you must remember a couple of things about this, novel devices to collect samples at home are making it possible for people to have easier access to medicine, to get diagnostic or to get advice without necessarily leaving their home. And to do that, we need to be able to compare one individual to the normal range, the reference range. And so we need to combine those devices with these reference ranges to be able to have these patient-centric results. So that's the overall introduction of 5P medicine, and now I'll go through a handful of examples, many of which are taken from my podcast, The Metabolomist, where I have the chance to talk with other scientists who are explaining their work to us. So I will show you a number of studies, and if you like those studies, if you want to learn more about them, you can go directly to the episode where I discuss it with the scientists, and they can tell you themselves more about that story. So our first P is preventive medicine. And here the example that I put forward is one that I discussed with Cristina Leguido-Quigley. And with her team, what they did is they had a cohort of children and adolescents that they investigate, who have obesity, who are affected by obesity. And they first used lipidomics to compare their lipidomic profile to a group of children and adolescents with normal weight, to look at what that looked like in terms of lipid profiles. And what they found, without too much surprise, is differences in triglycerides, in glycerophospholipids and sphingolipids, which are typically the kind of complex lipids you would expect to be different in the blood of people with obesity versus people with normal weight. But what was really interesting for me in this study is that they went to look at new ways of preventing or let's say restoring weight towards the normal range and preventing not so much obesity, but the diseases that we know are influenced by obesity. And so what they did here is they designed personalized intervention strategies. So this was a mixture of diet, but other lifestyle changes that were implemented for a year in these children and adolescents. And then after that, they measured again the lipidomic profile, and they could see that the levels of those complex lipids were getting very close then and changing drastically and getting closer to those of the children with normal weight, which is very encouraging for both, using omics in general, I would say, but especially these very sensitive, flexible, reversible omics that are metabolomics and lipidomics, to first get a picture of what the disease looks like and also be able to monitor whether that picture moves back to normal state. So I really encourage you to listen to that episode. It's from this season of the podcast, and really an interesting an interesting topic. Second, for predictive medicine, I have here an example focused on mental health. So in this case, I discussed with Sabine Bahn the use of her group of metabolomics to discriminate between bipolar disorder and depression or unipolar depression. And these are illnesses that are very difficult to discriminate because still today there is no physical measure of those illnesses. So diagnostics and also the choice of treatment is based on questionnaires that are both a little difficult to have really pinpointing and separating these pathologies, and also very strenuous for the patients to take. And it's difficult when you have these diseases to actually really focus and answer these questionnaires in an efficient way, and it's really painful for the patients. The problem is then if you have wrong diagnosis, you also get the wrong treatment, and it's already quite difficult to treat patients with, especially with depression. So whatever we can do to make it quicker and more efficient to have diagnosis of these illnesses is really a high value solution. So what this team did here is they applied metabolomics and they found a signature of 15 to 20 metabolites that could discriminate relatively well those two pathologies. And when they combined that with answers to parts of the questionnaire, they had an even better separation. I'm not going to go into the detail of the signature, but you can just listen to my discussion with Sabine Bahn about this on the podcast. What's really interesting for me is that several of the small molecules that are part of that signature are actually microbiome metabolites. So if you go back to what I was saying at the beginning, using microbiome is a really powerful way to look into this. Precision medicine is a tool that's very powerful for stratification. Here I have an example where I discussed with Rachel Kelly from Harvard Medical School the way that her group separated a group of children with asthma into 5 subgroups. And these were so metaboendotypes or metabotypes, and each group has a different pattern that is informative then for potential treatment options and original ways of addressing each patient or group of patients with the most effective treatment. The last example is related to population-based medicine. And here we have a case study, so from a publication by an Italian group, combining exome-wide association, an exome-wide association study with metabolomics. And the reason why I chose this example is that what they found is what I consider the main thing that people try to achieve when they combine a form of genomics with metabolomics, is to find new variants that are associated with the traits that they're studying that they wouldn't find with genomics alone. So what's interesting here, for example, is they had 39 variants that were not previously identified. And these associations with metabolomics are not random. So 12 of those were associations between the enzyme and the metabolites that they directly process. 12 others were enzymes in the metabolic pathways of the metabolites they associated with. And so this shows you that you can, combining different omics, you can really start to highlight pathways that are the ones you're going to want to focus on later on.
One minute, please.
Yeah, I'm almost finished. Thank you, Carmen. So the way forward with multiomics, the main aspect for me is you have to know how to analyze this data. So if you have a bioinformatician, this can be helpful, but they might not be experts at all omics. And if you have a piece of software that just processes everything automatically, you lose that kind of human touch that is needed, especially for the interpretation afterwards. So what we provide together with our partners at Eugene is a semi-automated pipeline that processes omics together. And we will hear a talk by the chief scientific officer from Eugene later in the webinar, and I encourage you to listen to her take on how to do this in the most efficient way. But in the end, for your own study, I think the best combination of omics always depends on your goals. So depending on what you're looking at, this is how you pick the omics, this is how you pick the time points or the conditions you're going to compare. And if you're looking for inspiration on how to do this, I point you to my book, The Story Principle, where I discuss how to design metabolomics and omics studies and give some tips there. Thank you very much for your attention.